हाँ
ठीक है।
यस मैम प्लीज स्टार्ट।
Okay, thank you, ma'am. Good afternoon, respected Dr. Dongarwar sir, Dr. Preeti madam, my teacher, Dr. Hate sir, Dr. Borkar sir, Dr. Bansod sir, and my dear friends. I am Dr. Lalita Savarkar, Assistant Professor in Botany in KZS Science College, Brahmani, Karmeshwar. Friends, I am presenting here a seminar on algae and its application. That means, what are the uses of these algae in our life? We all are aware about this topic because it is already included in our syllabus. Okay, but still I like this topic, so I am presenting here. The term algae cover many different organisms capable of producing oxygen through the photosynthesis. It means that the process of harvesting a light energy from sun and it converted into the carbohydrates. These organisms, that is algae, it's similar to the higher plant in some character, but it's still different in some structure also. Madam, next. If we see the algae is thalloid body. It means that it is not differentiated into root, stem, and leaves as a higher plants. Second difference is that the algae does not have any kind of vascular system for the conduction of water and minerals, but vascular system is already present in the higher plants. Secondly, algae is present in a different form and size. The algae may be unicellular, single cell, microscopic, macroscopic, filamentous, colonial, branch, or it may form a leafy appearance just like the seaweeds. Okay, and it may shows the size of 60 meter in or more than this also in the length. Algae mostly found in aquatic habitat. It shows two kind of habitat or two kind of water sources, fresh water and salt water. That means marine environment. Algae are also able to survive on the land. Some unexceptional locations are there where we can found algae, just like a tree trunk, animal fur, hot springs. Madam, next. Uh, There are different kinds of algae are present in environment in the nature on the basis of their pigment system, on the basis of their structure. There are different kinds of algae are there. Blue-green algae, that is Cyanophyceae, then Chlorophyceae, Phaeophyceae, Rhodophyceae, diatoms, that means Basilorophyta. There are different kinds of algae are present in this world. I'm mainly focusing on the topic ecological and commercial importance of this algae. So first application of this algae is, as algae is a plant, it belongs to the plant kingdom. They have the chlorophyll, so they can produce a food by the process of photosynthesis. It is a main producer of a food chain. In the ocean or in a marine environment, algae are present at the base of food chain. Many of the marine life includes whales, fishes, turtle, sea stars, worms, are ultimately depends upon the algae for their food. Second application is a production of oxygen. We know that each and every organism or a plant जो photosynthesis कर सकते हैं, they will produce oxygen as a byproduct. Algae produce an estimated 30 to 50% of net global oxygen, which is available for all the organism, all the terrestrial organism, animals for the respiration. Third application of algae is a food, and it is a wide application. Most of the region of the world use algae as a food in processed form or unprocessed form. In East Asia and Pacific Island, algae directly consumed as a food. Many countries grow algae for food and earn a billions dollars from it. Red alga, just like porphyra, is a most important commercial food algae. Second, uh, dulce, that is Palmeria palmata, is also one of the red algae, another red algae which is used in Canada, US, and Scotland. Then Laminaria is a brown algae. It can be harvested by the hand from the rocks during low tide. And this algae, Laminaria algae, is used in Japan, Korea, China, where they may be eaten with the meat or fishes or in the form of soup. Green algae, just like alva, are eaten as a salad or in the form of soup, or it may be eaten with the meat or fish. Pure green alga, just like chlorella, is cultivated as a food supplement and is eaten in Taiwan, Japan, Malaysia, Philippines as it shows a high protein content. And it is a use for space traveler. It is used as a food for the space traveler also. Then this is a third application, that is, madam, next kara. Agar, we know that in a tissue culture technique, for the preparation of media, we use agar powder. And this agar powder is also extracted from the red algae such as Gelidium, Gracilaria. These are the red algae from which we can extract this agar-agar powder and which can be used for tissue culture technology. Then we can also extract one of the another polysaccharide, that is carrageenan, which are also extracted from the algae. For example, Chondrus is the name of the red algae from which we can extract these carrageenans, and this can be used for the thickening and stabilizing as a for the dairy product. So in this way we can use algae in a different way. Then we can use algae for medicinal purposes also. Algae is also used as a medical important sources because it has antioxidant, anti-cancer, antiviral properties. So this algae can have the different medical property. Then anti-cancerous algae, most of the diatoms algae, marine algae, and includes diatoms, most of the have the property of anti-cancer. Okay. Then there are different algae, just like chlorella, nostoc, laminaria, alva, fucus. These are some algae which belongs to the different groups. Some belongs to the cyanophyceae, some belongs to chlorophyceae, and some belongs to the brown algae. That means pyophyceae. They have the property of antiviral. Okay, they can be work on different virus, HIV, HPV, HRV. So in this way, there are different viruses. Okay. Then the most important and a recent application of these algae, and this was my that is the use of algae as a fuel. Actually, this was my PhD topic, so it was very interesting for me. So algal cells contain a lipid, hence this lipid means fatty acid can be converted into a ester, that means a biodiesel form, by using some catalyst and some other reactant. So algae can be used as a fuel. In the literature, I just when I go through the literature, I found that the Botryococcus is one of the algae which grown in on the lakes, and this Botryococcus release some oil or produce some oil, and this on the water surface, this oil just extracted or collected by some companies and used as a fuel. I actually don't know exactly what is the process or what. I just go to the literature. I thought it is new. That's why I shared with you. So this is also one of the fuel. Up to this, we have seen just positive approaches of these algae, but there are some negative approaches also. That is the toxicity. What happens when the algae grown in any area luxuriantly? Some of the algae, just like the dinoflagellates, produce some toxins, and if some animals are there, some aquatic animals are there, they consume this water and they may die. If we also we may consume this water and we may get suffer from vomiting or some abdominal pain. So in this way, there are some negative aspect of this algae also. Madam, next. Thank you.
Thank you,
Thank
Dr.
you, ma'am.
Lalita ma'am. Yes. Okay.
वेळ झालं ना?
Yes, yes, no issues.
Okay.
Thank you. Thank you. Next one is Dr. Shailesh sir, Shailesh Bansod.
Yes, yes, madam.
Yeah. Yes, sir. Can you
Hello.
see?
Yes, ma'am.
Yes, please start.
Yeah. Good afternoon, all. Respected Dr. Dongarwar sir, Dr. K. Madam, and dear friends. Myself, Dr. Shailesh Bansod, Assistant Professor, Department of Botany, Arvind Babu Deshmukh Mahavidyalaya, Barsingi. So today I will be delivering a seminar on biometals by algae. Now, next slide, madam. Hello. Hello.
Yes, sir. Yes.
Sir, it is
Yes.
changed.
Yes. So we know that so many industries is growing day by day. That means the industrialization in large amount takes place, and due to the anthropogenic activities, so that there is a discharge of waste and wastewater which contains heavy metals, and it is being released to the environment. Now, what are the possible sources of heavy metals? According to Oleski, he analyzed some sources of heavy metals like mine draining, metal industries, petroleum, refining, tanning, electroplating, etc. Now, we cannot ignore the pollution caused by the heavy metal, because environmental contamination due to heavy metals can be a serious problem because their incremental accumulation in the food chain. So that's why we cannot ignore the contamination of heavy metals in every in ecosystem. So there are so many technologies have been put forth for the removal of heavy metal. So conventional technologies that were given like a chemical precipitation, filtration, chemical oxidation, reduction, and reverse osmosis, ion exchange, adsorption, and evaporation. Now, what is the drawback of all these technologies? They were not found to be effective or prohibitively expensive, and also inadequate in case of low metal ion concentration. It means even the heavy metal concentration is low, so these technologies are not able to remove that heavy metal concentration. So there is an urgent need of environment friendly technologies should be developed in order to remove heavy metals from the wastewater. So the concept of biosorption is more emphasized after the use of conventional technology. Now, this biosorption technology is promises to fulfill the requirements. So, this biosorption technology is very competitive, effective, and cheap. Now, we will see what is biosorption process. Biosorption, we know that, and it is just like a adsorption, but instead of using synthetic material, so we are using here the biological material which is environment friendly. Okay, so biosorption is a process that utilizes inexpensive dead biomass to sequester toxic heavy metals. So the removal of heavy metals by using some biological organism, maybe in the form of living or dead material. Okay, so these organisms used in the form of living or non dead material that can be called as biosorbent, because the materials is biological material. So like bacteria, fungi, and other algae of marine origin. Marine origin, you will find marine algae like sargassum. Recently, Dr. Lalita uh, explained the marine algae also. Okay, so that can be used as a biosorption. So we will see next slide, ma'am. Next, madam, please next slide.
Sir, third slide are you talking about?
Yes, I already explained that one also. Yeah. So major advantage of biosorption process for conventional technologies or the methods of removing heavy metals. So this biosorption process is low cost. It is high efficiency and minimization of sludge production. See what happens in conventional technology, by removal of heavy metals can be produced a large amount of sludge. So if we use biosorption, there will be a minimization of sludge production. Okay. So whatever biosorbent we are using is regenerated. Means if we use the dead biomass and after the biosorption process, we can separate heavy metals and the biosorbent. So we can again use it, that biosorbent, for the process. So here there is a possibility of metal recovery also. So all these are the advantages of biosorption process. Now we will see what are the factors. Madam, next slide. We will see the biosorption process. What are the influencing factors? The pH could be the main governing factor, because this process requires a specific pH for the metal adsorption. Okay. Now temperature. If this process, second parameter is temperature. So the biosorption process can be of great concern in the range of 20 to 35 degrees centigrade. Now, third one is the biomass loading. So, low biomass can uptake more heavy metals, but there is a competition among the heavy metals. So, biomass should be optimized. How much amount of biomass we have to load in the process is very important. Then next one is the presence of other metal ions. So, if you want to remove one heavy metal, but in that solution or in that wastewater, other metals are also present. So there may be a chances of competition among the metals. So all these are the factors which influence the biosorption process. Next slide, ma'am. Now we will see what are the materials and methods which is required to perform this biosorption. So one by one we will see the materials. So first of all, you require biosorbents. So you may use dead biomass of algae, dead biomass of fungi also, and some agricultural waste we can use as a biosorbent. Okay, but after selection of biosorbent, that biosorbent should be pre-treated. Okay, so if any impurities is present, we can remove that impurities prior to the batch mode studies. Okay, now what which metal you are going to study? So the stock solution of that heavy metal ion should be prepared. Now this is the materials. Now coming to the part of methods. So that includes a proper biosorption studies. So in biosorption studies, we need glassware and apparatus. So generally this process is carried out in the Erlenmeyer flask of 150 or 250 ml. And we require some orbital shaker, pH meter, etc. So many things are required for that. So after that, actual process, before starting actual process, it should be optimized. Yeah, I already told that the influencing factor like pH, temperature, biomass loading. So we have to minimize or optimize the effect of pH. Then second is the contact time, how much time it will take heavy metals to adsorb. Okay, so that should be optimized. And at what temperature the heavy metal is again going to adsorb, that temperature we have to optimize. Then biosorbent quantity, whether it require 1 gram, 2 gram, 3 gram, 4 gram, etc. Okay, that should be optimized. And how much amount of that metal ion concentration we require to carry out the biosorption process. So that should be noted. Okay. Now, after getting data of all those things, so next part is equilibrium isotherm modeling, because this is the adsorption thema, that is the uh, chemistry phenomena. Okay, so there are certain adsorption isotherm that we have to check, like Langmuir adsorption isotherm, Freundlich adsorption isotherm, and we have to fit our obtained data to these adsorption isotherm. So on on that basis, we will be able to state that Langmuir is fitted or friendly adsorption isotherm is fitted for the data. Then next is we have to carry out kinetic studies, which includes the order of reaction. So whatever the experiment we have done, whether it is going to follow the pseudo second order model or first order, etc. Okay, so that should be optimized. So these are the material methods we require for the biosorption process. Next, ma'am. So how to study the mechanism of biosorption? If anybody says that this cadmium is adsorbed on this particle, but how you will come to know? Because being a science student, we should provide explanation and proofs for that. So in order to study the mechanism of biosorption, scanning electron microscopy is used. Then transmission electron microscopy, that is also used. Field emission scanning electron microscopy is used. So by using all these techniques, we can study the mechanism or characterization of particular heavy metal molecules, whether it is adsorbed on the material or not. Okay, next slide, ma'am. So coming to the main part. So by doing this biosorption process, so many algal material in the form of living or dead biomass is used as a biosorbent. So this table indicates different algal species which is used as a living, that is the active or inactive form, like Durbelia, Iplonia, Homocera, Chlorella virgalis, क्लोरेला वल्गैरिस कॉपर कॉपर के लिए उसको यूज किया है. देन सिनेडेस्मस abundance. So this सिनेडेस्मस abundance is used in the both form, active and inactive. So active form for copper removal and inactive form for cadmium removal. So like microspora, sargassum, some Theophysian members like seaweeds, they were also used. So extensive studies was carried on these algal species, and they were found a very effective one on the eradication or the removal of this cadmium, chromium, zinc, lead, etc., and nickel too. Okay, so these tables. Next slide, ma'am. So here also you will see the different algal species in the form of in the active or inactive form, and the metals which they are removed, and these are the references or the authors which carried out the studies on the algae. Next slide, ma'am. So here is also some cyanobacterial studies on some cyanobacteria like Anamina, Microcystis, Eudorina. So it is also given. Next slide, ma'am. Now we are using algae as a biosorbent, but what characteristic feature it has or the some biochemical agents which the cell wall of these algae is having, like cellulose and peptidoglycans and special polysaccharides. So these are present on the algal cell wall, which provides some chemical groups for the heavy metal binding. Like in Pheophycean members, algine is present, which provide amino carboxyl group and sulfate group for the heavy metals to adsorb. Okay, so what are the important things present in the algae? That is the metal ion binding sites. So these are the cellulose, peptidoglycans, special polysaccharides. Then in Pheophyce, algin, it provides so many groups to that heavy metals to adsorb. Next slide, ma'am. Now these are some functional groups which are responsible for the uh, metal biosorption. Like some basic group is there: amino, alamino, cyclic, non-cyclic, carbonyl, ether, alcohol, and some acidic group like carboxylic, sulfonic, phosphonic, phenolic, and phenolic, etc. Next slide, madam. Now we have carried out some study on the manganese mine water. So under the able guidance of late Professor P.B. Nankar sir was my guide and an eminent algalogist from the central India. So I carried out this study on the four manganese mines from the Vidarbha region, like Gumgaon manganese mine, Kandri, Mansar, and Beldongri manganese mine. So we surveyed these mines. Next slide, ma'am. We surveyed these mines, collected the mine water, we analyzed the heavy metal contents on the ICP-AES and AS. So we got these heavy metal concentration. So as far as these mines are concerned, they are the manganese mines. So these water mostly contain uh, iron and manganese. Because wherever there is a presence of manganese, so iron is there. So we will be concerning on the Fe and Mn, ferrous and manganese. Next slide, madam. So we surveyed so some of the algal material present on the uh, on the surface of water in the form of mats. So we have collected Udogonium glabrum as shown in the slide from the uh, mine waters collected from the main open cast mine. Okay, then Spirogyra virucosa, then Microspora flocosa. So all we carried out studies of biosorption by using these three algal species. Next slide, madam. Now, as I mentioned, how to carry out these biosorption studies. So according to the material methods which we follow, we optimize the pH, we optimize the contact time, we optimize the temperature. Okay, so all these studies were performed. Then the percent removal and the amount of metal ion concentration required, that has been also carried out. Now, regarding pseudo second order reaction, so chemical kinetics was also studied. So mostly the second order reaction. See here in the table, we got R square values. Next slide, madam. See, this is the graph showing the pseudo second order kinetic plot, and the table shows R square. If you have look at the R square value, that is above zero point 0.9, it indicates that this reaction follows pseudo second order reaction. Next slide, madam.
Sir, your time is up. Kindly sum up.
Madam, only two slides.
Yeah.
Then Freundlich adsorption isotherm, we have studied Langmuir adsorption isotherm. So in all isotherm, the R square values are above 0.9. So this suggested that the suitability of all these adsorption isotherm. Next slide, ma'am. Next slide, ma'am. This is the Freundlich adsorption isotherm. So this is also suitable for the bioabsorption study by the algal species. Next slide, ma'am. So this is the overall data we have represented in the form of table. Next. Next slide, ma'am. Now, conclusion. These three algal species where that can be employed as a low-cost and eco-friendly biosorbent for the removal of heavy metals like Mn and Fe from the mine water. So, this is all about the small case study. Thank you. Thank you, ma'am. Hello. Hello.
Nice presentation, sir.
Hello.
Yes, sir.
बोल
Yes.
शैलेश.
Good afternoon, sir.
Yes.
Sir, nice presentation, sir. Dr. Sangode बात कर sir.
Okay.
Okay. Thank you.
I request everyone to be please stick to time because we have to complete all the seminars on the scheduled date only.
Thank you, Dr. Shailesh, sir.
Thank you, ma'am.
Yeah. Sir, काही questions विचारायचे आहेत का डोंगरवार sir? Are you there?
नाही मॅडम.
Okay. Proceed
Yes,
कर.
we have some
Yeah.
question.
Yes, participants आपको पूछना है.
No, madam.
Okay. Fine.
Madam, I am audible. I want to ask question.
Yes, yes, please.
Sir, excuse me, sir.
Yes, ma'am.
सर आपने इसके बाद और किसी पे काम किया है? माइंस पे ही किया है, लेकिन और कोई सॉइल पे, पॉल्यूटेड सॉइल पे किया है कहाँ?
Yes, that is, madam, which I represented here a small case study because due to lack of time, this is one of
Okay.
the part of my thesis. So I have studied soil also, mine spoil dump. Basically, my topic was ecophysiological studies on algae associated with mines of Vidarbha. So in that, I have studied the mine water, then water quality index for the social constraint. Okay. So, and also the indicator species of mining or mine water. So all these things I have studied.
Okay. Thank you. Very very informative work, sir.
Thank you.
Thank you, Shailesh, sir. Next participant is Dr. Naina, madam. Naina Shirbhate.
Yes, ma'am, I am here.
Yes. Yeah. Presentation this time.
Oh,
Yes. Please start.
okay, ma'am. Thank you. Good afternoon to respected Dr. Dongarwal, sir, Dr. Dharmik, ma'am, my guru, Dr. Vinod Chauhan, sir, Hate, sir, and all faculty members of various colleges. I am Dr. Naina Shirbhate from Vidyavikas Arts, Commerce and Science College, Samudrapur, going to present on topic environmental impacts of idol immersion. Next one, ma'am. Ganesh festival is celebrated traditionally in the state of Maharashtra in a big way as a social and community activity. It was started by Lokmanya Tilak in 1896 at Pune. The main objective of the festival was mainly to bring people together and promote freedom movement. But nowadays, this picture is totally changed. Next one, ma'am. Next one. And select Amravati city for my study. Population of Amravati city is now about 6,50,000 and available water sources from Appavada Dam, tube lakes and various rivers. Ganesh festival celebrate every year in months of August or September. That's why I'm select this topic because nowadays Ganpati and Ma Durga festival in this month we celebrate these festivals. 3,000 to 5,000 adults of Lord Ganesha of various shapes and sizes were immersed in Chhatri Lake located outskirts of Amravati city. Please, ma'am. Next one. Next one, ma'am. This is the site I am selected, which is situated. This is the Chhatri Lake, situated near MIDC Bypass Road, Amravati, outskirts of the Amravati city. Next one. Next one, please. And select samples from that site before and after adol immersions. Next one. Next one, ma'am. What is this? In what way we immerse this idols into the lake water or different water bodies? Next one. And what are we doing? We worship goddess uh, Durga, Ma Durga, and Lord Sri Ganesha. And in what way we immerse that idols in water bodies? Next one. Aims and objectives, and collect water sample from that site and analyze different parameters, that is the temperature, pH, dissolved oxygen, BOD, and alkalinity, the chloride contents, etc. Next one. Next one. And found this result. Mostly the dissolved oxygen level is lowered down after the immersion. Then with biological oxygen demand, BOD level is rises. Then conductivity, salinity, then different chlorides. These parameters are rises 20 to 30 folds higher after the idle immersion. Then next one. The important parameter in water bodies, that is the dissolved oxygen level. The dissolved oxygen level is very lowered down after the idle immersion. I am sampling in both before idle immersion and after idle immersion. Both I am found differences the levels of dissolved oxygen. Next one. Next one. Affects the pollution level of Chhatri Lake and different water bodies when we immerse the idols of Lord Ganesha and Ma Durga. It's been increased year after year due to the increase these types of activities. People use lake water for irrigation, drinking, and bathing of animals. And different physicochemical parameter affect on aquatic flora and fauna. Next one, ma'am, please. Uh, Adequate dissolved oxygen is necessary for good water quality. Dissolved oxygen drop down below 5 milligram per liter due to the aquatic life under stress, and high chloride content impact taste and odor. It could cause corrosion. Next one. Next one, ma'am. Effects in this lake water, it affect on the animals, then flora and fauna under the stress, and this water is used for the irrigation. It ultimately affect on health of human beings or different animals or other fauna. Next one. Next one, ma'am. A conclusion, all the physicochemical parameters of Chhatri Lake recorded after immersion of idols were 20 to 30-fold higher than the before immersion. Then changes in oxygen temperature and pH, it affect on the chemistry of the water after triggering the chemical reaction, resulting in the formation of unwanted products in that water bodies. This indicates serious environmental issue. In order to protect water bodies for this state, mass public awareness is needed. Besides pollution control authorities, local peoples should work hands in hand to combat against this dangerous environmental problem. And immersion violates provision of Water Prevention Pollution Control Act 1915 to Environmental Protection Act in 1964. Next one. And lastly, thank you. These are some possible solutions. Use eco-friendly idols made by the natural clay and paper pulp. Use natural dates for painting. Immersing the idols in water tank in a bucket of water at home, and avoid the use of different decorative material used at during the festivals. That is the thermocol sheets, polythene bags, etc. And after the immersion of the idols, in particular artificial tanks or other water bodies, plantation done at the immersion site is very important. Next one. And thank you. Thank you very much. Next one, ma'am. Next one. These are some references. Next one. Thank you.
Thank you, madam. Madam, what is your subject? Botany.
Botany. Yes, ma'am.
Okay. Why you have chosen this topic?
This topic is environment related, and I do my PhD on environmental based topics, that is the phytoremediation of bodies as well as the soil.
Okay. Sir, any question?
Uh, have you noticed any changes in the vegetation before and after the idle immersion?
Yes, sir. Different varieties of plant found, just like the Ecornia hydrilla. It's developed in many times. These plants have ability to absorb some metals or different types of ions, and these plants grow well that conditions.
Okay. And does Amravati Municipal Corporation allows household Ganpati idols to be immersed in the water bodies? Because in
Now,
Nagpur,
banks
yeah,
are
in Nagpur they don't allow. Household Ganpatis are
now
not allowed. Yeah.
मंडळ जे असतात ना ते इमर्स करतात सर त्याच्यामध्ये अजूनही.
Okay, madam. Thank you. Are there any
Hello.
questions from the participants?
Yes, sir.
Yeah.
Ma'am, actually, I just want to know exactly what is biological oxygen demand with respect to the polluted water, and how why you had shown that it is going to be quite race much with respect to the graph shown over there. Will you justify the relationship, please?
Okay, in water bodies having different faunas, different animals. These are under stress. They require amount of water, amount of oxygen for their survival, and dissolved oxygen level getting lowered down after the immersion of Ganpati idols. That's why I'm focusing on that graph.
So, ma'am, you mean to say if we immerse a POP or even the kind of clay onto the soil, automatically it is having a chemical reaction with respect to H2O, and the level of O2 is
This
going down, or in
P-
terms of what?
POP, or we decorate these idols in different. Okay, we use different dyes,
Like
artificial
paints and all,
dyes.
na?
Huh. And these dyes contain different heavy metals and different ions. Okay, that rises some chemical property triggering chemical changes inside the water bodies. It ultimately affect on the different physicochemical properties of that water bodies. Okay, sir.
Okay, okay, ma'am.
Okay. Thank you, Naina madam. Next
Thank you.
participant, Dr. Jagannath Gadpaile sir.
जी मैडम.
हाँ, PPT
Am I
दिसते
audible?
आहे? Yes, you are audible.
Yeah.
Yeah, please start.
Thank you, ma'am. Respected Dungarwar sir and Priti ma'am. I am Dr. J.V. Gadpaile, Assistant Professor, Department of Botany, SN Moore College, Tumsar. I am going to present my presentation as a seminar topic for this life refresher course in life sciences. So my topic is Saida species from Bhandara district, Maharashtra state, India. Why I choose this topic? During this refresher course, I found that there are some speakers, respected Chaturvedi madam, then respected Barsagade sir and Uma Thakur madam. They also focus on some conservation issue. Invasive plant, Dr. Chaturvedi madam, then Uma Thakur madam focus on the conservation of orchid plants, and today's speaker, Dr. D.D. Barsagade sir, explained that the plants are important for the biodiversity. Any biodiversity may be because that may be not, that must be because of the plants. So I'm working in Bhandara district for the floristic study from 2015 and before that. So I'm collecting the plants and identifying the plants and preparing the small reports regarding the taxonomic and systematic study of that particular plants. So I'm covering the Bhandara district regarding the angiospermic plant, and that means the floristic work of Bhandara district. So during my floristic visit to the various places throughout the Bhandara district, I found that there are some interesting species of Sida, And after that, I go through the references and the available literature. And after that, I found that there is some lack of, or you can say the least concern to the species of the Saida throughout the Bhandara district. So this one is the first slide, which represent the total status of the Saida throughout the world. There are 250 species are distributed in the tropical and subtropical region, particularly in the New World, tropics and Australia. In India, it is represented by 22 species, and the recent work that was given by, or that was published by the Gawande sir, then Sivarajana Pradeep and the Tambe sir. So that species, and due to this, the 26 species, because of the 6 endemic species are in India of the Saida. Throughout the Maharashtra, there are 14 species till today reported, including the few new reports, just like Saida Sivrajni, that was done by the Tambe sir and the Sardesai sir. While working in Bhandara district, I found that there are different kind of interesting species of Saida, and then I go through the literature and then collect the samples, prepare the herbariums, and then make it convenient for the identification. I communicate the expert, and after that, I found that there are 10 species. Previous studies says that there are only 5 species reported from the Bhandara district. After that, I am adding, after this study, I am adding the 5 more species to the Bhandara district floristic well. Next slide, ma'am. This is the site where I work throughout the Bhandara district. I work for the floristic work, and this is the satellite map, and the complete work is completed in the Bhandara district. Different kind of localities I visited, and collect the specimens, and identify that on my own best, as well as with the help of expert. Next slide, ma'am. So during identification of the Sida species, there is a requirement to find out the actual characteristic of the Mericarp. If you are able to diagnose the Mericarp characteristic in Sida species, then and then you can identify which one is the species, because there is some morphological resemblance in the species, and we can't identify the actual that species of the Sida. In that case, you have to dissect the fruits, and then you will get the Mericarp, and after observation of the Mericarp, you are able to find out which one is that the complete, which one is that species. So these are the structure from different Sida species, and I would like to acknowledge here Dr. Tambe sir. They permit me to take this slide for your presentation. Next slide, ma'am. These are the structures of Mericarp in the Saida. This one also, these are the different kind of Mericarp found in the different species during their study of the Maharashtra. Next slide, ma'am. So these are the species I collect from the Bhandara district. First one, that is Saida alnifolia. This is the new record for the Bhandara district. During my regular visit, I found that this plant species mostly found along the roadside, as well as in the cultivated area as a weed. The flowering period of this plant species August to February, and it grows throughout the Bhandara and as well as along the roadside luxuriantly. There is a huge population of this species. When I observed this, then I found that it is different. And after the Mericarp study, I found that this plant species is the Sida alnifolia. So this is one of the species which is new to the Bhandara district. New, ma'am. Next, ma'am. This is Sida acuta species. This is the common throughout the Maharashtra, throughout the India. This species is common, and most of the species, most of one from you, you are able to find this. And this plant species in mostly Marathi known as a chikna. So Sida species in Sanskrit, that is called as bala. Then in Marathi it is the chikna. So this one is the plant species. Tribal people and the farmers collect this plant species and make their some, you can say in Marathi, jhadu. So they are preparing the some tools for the cleaning process. Next one. This one is the Sida spinosa. Previously, this species reported by the previous taxonomies throughout the Bhandara district. They report some kind of Sida species, that is 4 species. So Sida spinosa, that is one of the common species found in Bhandara district. It looks a resemblance with the you can say the Sida angustifolia, but the leaves are different in angustifolia and spinosa. There is totally different. I will go to the next slide. You will find the what is the difference between Sida spinosa and Sida angustifolia. Next slide, ma'am. This is Sida cordifolia. This plant species mostly found on the open places and near to the water bodies along west area. This plant shows the different kind of mericarp in its fruits. You found that there is horns. They are very much larger than the rest of the species of Sida. And when you cut that plant or you just hold that plant, then the horns may or might be a chance to penetrate in your hands. So during collection of this plant species, you must be take precautions for the collection of this plant. These horns are very pointed and maybe penetrate your hands. And the flowering period of this plant April to November. This is Sida pardifolia. Next slide now. This is Sida cordata. This is the common species. It is mostly found nearby the house, or you can say the open area where mostly there is untouched area, or we can say along the roadside westland, and there is this plant is found. And so many people know this plant, that is Sida cordata. And flowering period, that is August to January, and distribution found open places along roadside and in westland. Next slide, ma'am. This is the another kind of you can say this is new to the Maharashtra, that is Sida, sorry, Bhandara district, that is Sida rombifolia. And this work represent the addition to the Bhandara district. Flowering period of this plant, that is from September to January, and almost it is along the roadside. Next slide, ma'am. See the huge vegetation of this, that is Sida rhomboidea, a new addition to flora of Bhandara district. And the flowering period September to January, distribution found along the roadside under the shady places. So this plant is collected from the Mohadi Tehsil, and I found that mostly 4 to 6 habitat where there is a luxurious vegetation of this plant. So I took this plant and do the details under the microscope for the mericarp observation. Then it identify as a Sida rhomboidea. Next slide, ma'am. This is the new addition, and this one is the new species recorded and discovered by the Tambe, Sardesai, and Pandesar. And this plant species recently in previous year I collect this plant species from Varti, that is near to the Bhandara. And when I communicate with the Sardesai sir, then they ask for the mericarp. Then I show all the details, and then they identify this plant species is a Sida Sivrajini. Then they say that this report that this plant species new to the Bhandara as a new type locality for this species. So this is the important, and you can say this is the interesting species, because in this plant species we found that 3 different kind of leaves. At the best there is a rhomboidal type of leaves, ovate, and then as you just come upward, then there is a linear leaves. After that, the awns or mericarp structure is different from rest of the Sida species. So that's why it is different, and identify and give the name by these 3 legendary taxonomists as the Sida Sivrajani. Next, ma'am. This is the unusual species, and I think this plant species is not reported throughout the eastern Maharashtra still today. And in previous year, I collect this plant species from the Tumsar Taluka and then go for the detailed identification. Then I found that there is a berry crop which is totally different from the rest of the 22 species found throughout the India. And then I communicate with the expert, then they say that this is the Sida angustifolia. So this one is the plant species, that is Sidus plant species. And this is new report for the Eastern Maharashtra state from Bandara district. The new report, and it is communicated with the well-known journal. Recently, it may come in next month as a new report to the Bhandara districts. That is Sida angustifolia. Next, ma'am. So this is the summary of my work for the Sida species from Mandara district. Those are in white color. They are reported from Bandara district previously, just like Sida acuta, Sida cordata, Sida cordifolia, Sida mysurensis, and the Sida spinosa. While the yellow color, these are the completely new to the Bhandara district. That is Sida alnifolia, Sida angustifolia, then Sida rombifolia, Sida rhomboidea, and Sida sivrajini. So these are the five new reports to the Bhandara district. Throughout this my presentation, I conclude all these 10 species during my field exploration. Next, ma'am. I would like to thank Dr. Gajanan M. Tambe, Ramchandra D. Gore, and the Professor Milind M. Sardesai from Pune University for their continuous support and the help in identification and literature survey for this work. Thank you, ma'am. Next slide. This was the references, and this is the last one. Thank you, everyone, for kind and patiently listening.
Thank you. Thank you so much, Sir Gadpale, sir. Nice presentation and nice work, and nicely presented also. Wonderful.
Thank you, ma'am.
Dongarwar sir, any questions?
Congratulations, Dr. Jagannath.
Thank you, sir.
Nowadays, very few people dare to work in the field of taxonomy.
Thank you very much, sir. Your
Yes,
blessing are always with me.
my questions or a layman question is that why
Yes.
does the leaf of Sida rhombifolia is not rhomboid?
Sir, there is a variation in rhomboidea because due to it might be due to this some kind of nutrition.
See, in the photograph which you have shown in that rhomboidea shows rhomboid leaf, but the rhombifolia means it should have rhomboid leaf. But the this is at the identification level.
Yes,
I'm
sir.
just asking a layman's question. One
The,
thing that yeah, you have used the Mericarp criteria for identification, which is very clear because many of the times the people they get confused in Sida mysurensis, Sida cordifolia, and Sida cordata.
Yes, sir.
That's why Mericarp is very important for identification of the species.
Yes, sir. Yes, sir.
Are there any questions from the audience? If there are no questions, then next presenter.
Okay. Thank you, sir.
Okay, thank you. Next participant is Mr. Manmohan Baisare, sir.
Yes, madam.
Yeah. Yes, sir. Sir, PPT this
Yes, madam. Madam.
Yes, please start.
Yes, madam. Good afternoon, everybody. I speak on the topic of species level relationship in genus Iliocaris, family Cypressae. Myself is Manmohan Vaisare, Assistant Professor at NPW College, Lucknow. Madam, next slide. We discuss about the genus Iliocaris RBR with introduction, material and method
taxonomic character of plant, discussion and conclusion, and then reference. Madam, next slide. Madam, next slide. Introduction. Eleocharis is the cosmopolitan genus of family Cyperaceae. The species of Eleocharis RBR is less known in India, even in Maharashtra. The other aspect of the life history were subsequently studied by different workers, Kander in 1932, Chanag in 1931, Padde and Makde in 1982. The genus Eleocharis RBR appear to be natural based on the some anatomical criteria. Recluse in 1895 divided the
सर आवाज नहीं है.
Hello?
Preeti madam.
येस सर.
Hello?
येस येस सर. आता आवाज
Madam,
येतोय.
fifth slide.
ओके सर, आमच्या इकडलं कनेक्शन
हा
लॉस्ट
मॅडम
झालं होतं.
फिफ्थ slide.
येस. मटेरियल अँड मेथड.
Madam, fifth slide, madam, fifth slide. Wajud Khan MA in 215 in his book Cypress of Western Ghat, about 85 species of Cypress have been dealt in this flora. Following 09 species of Eleocharis distributed in Western Ghat, West Coast, and in central part of Maharashtra state. One is Eleocharis acutangula, second is Eleocharis atropurpura, third is Eleocharis congesta, fourth is Eleocharis dulcis, fifth is Eleocharis equisetina, sixth is Eleocharis geniculata, seven is Eleocharis mohammadi, and eight is Eleocharis retrophexa, nine is Eleocharis pyrilis. These species like Eleocharis Opugostites and Eleocharis swami, Eleocharis tetraketra are less known, but they are today difficult to found in the Maharashtra. Nantar second, next slide, madam. Madam, next slide. I am coming to the material and method. The present investigation deals with the study of species level relationship in Eleocharis of the family Cyperaceae on the basis of its taxonomic consideration. The taxa for this investigation were collected from Bhandara district of Maharashtra state. Provisional identification of collected plant was done with the help of flora of Presidency of Bombay in 1908. Sarma et al. Flora of Maharashtra in 1996. Species level relationship carried to the understanding the evolution of this unusual and biological significant genus Eleucharis RBR of Cyperaceae family. Next, madam, next slide. Madam, next slide. Author collected these 06 Eleucharis RBR plant. First is the Eleucharis geniculata. Second is Eleucharis atropurpura. Third is Eleucharis acutangula. Fourth is Eleucharis retroflexa. Fifth is Eleucharis dulcis. And sixth is Eleucharis setifolia. The morphological and taxonomical character of this plant is vulgar, and they are differ due to the different elevation range. Their stem architecture are also different. Taxonomical character is found on the basis of biome, stem, leaf, bracts, root, rhizome, nut. They can't discuss in a short time. So the time consumption, we come to the discussion and conclusion. Madam, 10 slide, madam. We come to the discussion and conclusion. 10th slide. From the taxonomic character found in plant Eleucharis, plant species of Eleucharis geniculata, Eleucharis atropurpura is terrestrial, and Eleucharis acutangula, Eleucharis dulcis is fully aquatic freshwater, and Eleucharis chitaria and Eleucharis setifolia is amphibians in habitat. Root of Eleucharis geniculata, Eleucharis atropurpura, and Eleucharis chitaria setifolia are fibrous, and root of acutangula and dulci is rhizomatous with the tuber or bulb. Water chestnut plant is leafless due to the absence of leaves. Photosynthesis in plant is carried out by the clump and stem. Flowers which are very small and occur on the tip of the clump. Stem of geniculata, atropurpura are triangular, straighted, erect, 2 to 20 cm tall. But the stem of Eleucharis acutangula and dulci, spongy and straighted, slender, 20 to 40 cm tall. In Eleucharis chitaria and Eleucharis setifolia, the stem is tough, slender, 2 to 8 cm tall. Eleucharis species show the variation in stem architecture. Leaves sit obliquely obtuse, glabrous, blade reduced in Eleucharis geniculata and Eleucharis atropurpura. Leaves sit flat, slender, acute, and membranous in Eleucharis acutangula and Eleucharis dulci. Leaves sit absent and blade reduced in Eleucharis chitaria and Eleucharis setifolia. Now, glum 3 to 6 boldly ovate, membranous, obtuse in Eleucharis geniculata and Eleucharis atropurpura. Oblong, ovate, loosely imbricate, keep extinct in acutangula and dulci. Bloom is compressed, elliptic, fertile, bloom upward in setifolia and Eleucharis setifolia. The this is the morphological and anatomical characteristics of the Eleucharis plant, 6 Eleucharis plant. From this data, the Eleucharis species are grouped. Next slide, madam.
काय म्हणतात भालेराव सर जिमला चालले का नाही तुम्ही
Kunal sir,
जिमला चालले
Kunal
का
sir, kindly mute yourself.
जात असतं का नाही 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 आता हे काय म्हणतात घरी गेल्यावर नाश्ताच करायचा आता
एक मिनिट
मी पण जवळपास
कुणाल सर
उपवास आहे का आज जिमला जाता का नाही नाही, उपवास असल्यावर
फक्त एकादशी असेल तर नाही भरपूर
हम्म.
डॉक्टर कुणाल
मी
तुरुकमाने लक्ष आहे का तुमचं
सर मी त्यांना मीटिंगमधनं एक्सपेल केलं हं, कारण की ते आपल्याकडे कॉन्सन्ट्रेशन
नाही
नव्हतं
नाही, बरोबर आहे,
त्यांचं.
बरोबर आहे.
हो ना?
त्यांचं लक्षच नसावं.
हां. आणि आपल्याला डिस्टर्बही होत होतं, तर मी त्यांना एक्सपेल
हो,
केलंय सर मीटिंगमधनं.
ठीक आहे, चालेल मॅडम.
भैसारे सर 1 मिनिट मी परत शेअर करते.
हा मॅडम.
हां सर, कंटिन्यू करा.
हा मॅडम, नेक्स्ट स्लाइड मॅडम. हा. From the morphological and anatomical data, from taxonomical data of Elocaris plant, the Elocaris arbiae species are grouped. Their character also intermingled to one another. These groups are evolved from different elevation range. Eliocaris arbiae include some extremely difficult species complexes. These indicate that the above supraspecific taxa are probably monophyletic. Monophyletic means they evolved in a separate life. Then Elocaris acutangula and Elocaris dulci have a rhizome tuber, but rhizomatous tuber is not found in Geniculata, Atropurpura, Chitaria, and Setifolia. They are mostly extraterrestrial in habit. Above all species of Elocaris show the same character like hypogeneous bristle, but in case of Chitaria setifolia, the hypogeneous bristle reduced to small 6-tooth cuff were called by Nij and Dix understood. The bipyramidal tree are several tooth gynophore of Scleria and many species of Fimbristalis. The stamens are entirely within the discs of Elocaris, entirely without the discs of Elocaris acutangula and Elocaris geniculata. The marshy and aquatic plant of Elocaris, like Elocaris chitaria, chitesi, similar to that of Elocaris geniculata and Atropurpura. On the other hand, plant is fully aquatic like Elocaris acutangula and Elocaris dulci, but differ in stem architecture in compared to Elocaris geniculata and Elocaris atropurpura, chitaria, also setifolia. The individual of this species of Elocaris geniculata and Elocaris atropurpura are placed as a sister to the Elocaris chitaria and setifolia. This group of species is differed by their morphological character, but Elocaris dulci is evolved from Elocaris acutangula. So Elocaris acutangula is a father of Elocaris dulci. Then next slide, I have given a reference of this topic, and thank you. Thank you, madam.
ओके, थँक यू
Madam,
सर.
next slide.
सर काही क्वेश्चन्स आहेत का डोंगरवार सर?
नाही मॅडम.
ओके.
Thank you.
May I? Hello.
Yes, yes, you can.
Okay. Dr. Bhaisare, very good presentation. My query is
Thank
one only. Okay. I am audible?
Thank you.
येस सर, येस यू
Hello.
आर
Thank you, sir.
ऑडिबल.
Yes, sir.
गडपाले सर,
Yes,
यू
sir.
प्लीज आस्क इन मराठी सो दॅट ही विल बी एबल टू आन्सर प्रॉपरली.
चालेल ना मॅडम.
येस.
सर तुम्ही तिथं एक सेटॅरिया स्पीसीज सांगितलेली आहे एलिओकॅरिसची, तर ती सेटॅरिया आहे की इक्विसेटिफोलिया आहे?
सीटेसी सर, सीटेसी आहे ती.
बरं, त्याच्यात दुसरं एक आहे ते मोनोफायलेटिक असं लिहिलेलं आहे की
हा.
एक प्लांट डल्सी फादर आहे आणि दुसरं त्याच्यापासून इव्हॉल्व झालेलं आहे. तर तुम्ही फायलोजेनेटिक वर्क केलेलं आहे का सर?
सर हे मॉर्फॉलॉजिकल अँड अनाटॉमिकल बेसिस कॅरेक्टर आहे, पण याच्या नंतर मी फायटोकेमिकल बेसिस वरती केलेलं होतं. तर तिथं पण सर त्याचे ते दोघांचे कॅरेक्टर आहे, डल्सीच्या आणि ॲकुटँगुलाचं ते सेम निघाले आणि त्याचं जे क्वांटिटी ऑफ केमिकल होतं ते पण सेम निघालं. त्याच्यामुळं जे फायलोजेनिक जे स्टडी आहे ते डिफरंट वर्कर केलं आहे, इंडियामध्ये नाही केलं आहे ते फॉरेनर कंट्रीमध्ये केलं आहे. त्याचं रेफरन्स घेऊन सर त्याला मी डल्सीला अॅकुटँगुलरचा फादर म्हटलाय. नाही,
अच्छा, तुम्ही
अॅकुटँगुलर
तिकडचे
डल्सीचा
ते
फादर
रेफरन्स
आहे.
घेऊन त्याला इनोमरेट केलेलं आहे. ओके.
हा.
हॅव यू साइट अरुण चांदोरेज रेफरन्स?
हॅलो सर.
अरुण चांदोरे सरांचं रेफरन्स घेतलं का?
चांदोरे सरांचं नाव नाही ऐकलं सर, पण मी कालकर सर आहेत भभूतीचे, आमगावचे जे भुजकुटे सर होते त्यांच्याकडे गेलो होतो मी खूपदा.
अच्छा अच्छा.
आणि आमगावचे ते सर आवडे सर आहेत त्यांच्याकडे गेलो होतो आणि
नाही नाही,
त्यांच्या
तुमचं
सगळ्यांच्या
एकदम चांगलं
माध्यमातून
काम आहे सर, एकदम व्यवस्थित काम आहे. तुम्हाला समोर जर काम करायचं असेल एलिओ कॅरिसवर, तर अरुण चांदोरे सरांना तुम्ही कॉन्टॅक्ट करा. त्यांनी पूर्ण इंडियाचं एलिओ कॅरिसवर काम केलेलं आहे. बस एवढंच.
ठीक आहे सर, त्यांच्या कॉन्टॅक्ट नंबर मला पाठवून द्याल.
बिलकुल बिलकुल पाठवून देईन सर. थँक्यू सर. थँक्यू
ठीक आहे.
मॅडम फॉर अलाउइंग मी.
Thank you so much. Next
थँक्यू
आहे
मॅडम.
संजय मेश्राम सर. संजय मेश्राम.
hello ma'am. I'm here.
येस.
Okay.
26 स्लाइड्स आहेत सर, थोडं
Shall I
लवकर
start,
कंप्लीट
ma'am?
कराल? येस सर, येस.
हो बिलकुल लवकर होईल मॅडम.
प्लीज स्टार्ट.
Respected my coordinator Dr. Nitin Dongwara sir and ma'am Dr. Preeti ma'am. Today's my topic is seed bank by tribal people. हे actually माझं वर्क नाही. This is not on only seed bank. Okay. No. Seed bank is, I think, we are very, very familiar, but basic concept of green revolution distinctly shifted the focus on Indian agriculture away from biodiversity to high yielding crop. This result is reduction the genetic base of traditional seed varieties that are the now on the verge of the extinction. आपण खूप प्रोडक्शनच्या मागे लागलो आणि खूप साऱ्या डायव्हर्सिटी ज्या आहे, सीड डायव्हर्सिटी, व्हाईट सीड्स प्लांट, ते आपण गमावत बसलो. Lockdown मध्ये specifically among the organic crops of farmer market, it is now the time the seed shine. Before the COVID-19, we saw more and more workshop on the seeding of seed farm, seed saving harmloom varieties, not just India but overall world. Okay. Then seed collection is basic concept in seed bank. These are the tribal peoples. Tribal peoples collect the seed from their native places, native forest, and hanging methods and drying methods of fruit they are using. These are the Swara tribes. They are collecting seeds, fruit, and then select these native methods of seeds. Uh, at the present, focus in more the seed multiplication and conservation. Last time, many farmers sowed the codes and felt the taken crop. So this time, code was grown only a small plots of land. मोस्ट ऑफ दी जो आदिवासी थे, इनका बेसिक था कि जो उनके पास है सीड्स, वो आगे चल के उसको सेव भी करे, उसको यूज भी करे, एंड देन जो है वो उनका यूज भी करते जब उसको संरक्षित करे. ठीक है. बेसिकली वी आर वेरी वेरी फैमिलियर दिस वर्ड सीड बैंक एंड वी आर बॉटनिस्ट, और एक बॉटनिस्ट के लैंग्वेज में कहना चाहता हूँ, सीड बैंक स्टोर सीड्स टू प्रिजर्व जेनेटिक डाइवर्सिटी and is a type of in bank or seed bank. The seed bank preserves dry seeds by storing them at a very low temperature. Spores and pteridophytes are converted in seed bank, but other seedless plants such as the tuber crop cannot be preserved this way. Okay. Scope हम देखना चाहते हैं. Basically common varieties, member state will collaborate with each other, development of list of common varieties, maintaining a seed qualities and seed varieties also. She is one of the best example, Punita Okebhai. She conserve many seeds in mud pots. This is the mud pot ज्याला आपण माडके म्हणत असतो. So this mud using by Punita Bai, she store many seeds, wild seeds as well as crop seeds. Process of seed storage in pots. बेसिक नेटिव प्लेसेस में जो बेसिक उनकी जो आइडियाज है, बेसिक उनके जो प्रोसेस है, उस प्रोसेस से वो सीड संरक्षित करते हैं। जो उनके पास बेसिक पॉट्स जो रेगुलर इसमें है, उसमें ही वो स्टोर करते हैं। दे आर यूजिंग अ वेरी प्रोसेसेस, ड्राइंग प्रोसेस, दे आर यूजिंग अ लोकल टूल्स, लोकल पॉट्स, लोकल जो कुछ उनके पास कंटेनर, बट they this local content made by special categories, special tribes, और उन tribes से बनाए जाते हैं। और वो जो है basically it's made by the plant, bamboo plant. Okay. uh, third slide, third photograph shows the drying techniques of seeds and fruits. Then the seed fruits and some कुछ ऐसे ये भी होते हैं rhizome, they are using a drying techniques, they are dry with the help of some fruit का। ये एक और प्रोसेस है जो घर के आंगण में स्कॅटर्ड फॉर्म में, ज्याला सडा टाकणं म्हणतात, फ्रुट्स एज वेल एज सीड्स, मोठमोठे असे सुप असतात बांबूचे बनलेले, उसमें उनका एक वर्ड है सड़ा टाकना, दैट मीन्स द स्कैटर्ड फॉर्म में वो सीड जो है ये सुखाते हैं। जनरली जो सीड्स है वो 12 आवर, एक के पहले सुखाते हैं, जो बहुत ज्यादा गर्मी होती है उसमें नहीं सुखाते हो। ये एक पीक है जिसमें ट्राइबल पीपल्स कलेक्टिंग फ्रूट फ्रॉम फॉरेस्ट, देन द फ्रूट उनको ड्राई करते हैं। आप सभी इनको जानते हो, पद्मश्री बीज माता जिसको हम कहते हैं, बीज माता, इन्होंने कई सारी बीज वैरायटी है जो संरक्षित की है, और उसको जो है संरक्षित करने के बाद कुछ बायोडाइवर्सिटी जो वाइल्ड सीड्स थी, वो वाइल्ड सीड को उन्होंने कंजर्व करके जो है संरक्षित करके जो एक्सटिंक्शन पोजीशन पे कुछ प्लांट्स थे उनको भी संरक्षित किया है। these are the tools using by tribal people, जिसको हम कूड़ी भी बोलते हैं, कोठी भी बोलते हैं, बट दिस कोठी मेड बाय द बेसिकली bamboos एंड सॉइल जो है इससे बनाते हैं। देन सॉइल से बनाने के बाद इसको त्याला लिपणं often served with the cow dung and neem oil, keep away insect and pest. Another method as use of the vino poti or bamboo structure, whether it's strong or the spread in the base of with the cow dung. पूरा उसको जो मिट्टी और काउ डंग से जो है कवर करते हैं, ड्राई करते हैं। देन जो है नेक्स्ट स्लाइड में जब वो सीड सिलेक्ट करते हैं, सीड सिलेक्ट करते समय जो है ड्राई एंड जो सीड यूजफुल है, जो डॉर्मेंट नहीं रहती है, वो सिलेक्ट करने की उनकी पर्सनल मेथड है, जिसमें वो सॉल्ट मेथड यूज करते हैं। लाइट एंड वेटेड सीड जो है उसमें डॉर्मेंसी शो नहीं करती, जो जर्मिनेट होने हो सकती है, वैसे सीड वो स्टोर करके रखते हैं, जिसको कोटी हम कहते हैं। नेक्स्ट स्लाइड में एक और है मेथड जिसमें एक तेलंगाना का है, जिसमें जायराबाद यहाँ पे कुछ जो टोकरियां जो बाम्बू से बनाई जाती है उसमें स्टोर सीड रखते हैं, देन रिकवर एश एंड देन काउडंग एंड मिट्टी, इससे बना के उसको कवर करते हैं। एश जो रहती है वो उन सीड को प्रोटेक्ट करती है। मोस्टली ग्राम वेराइटीज जिसमें रेड ग्राम हो, ग्रीन ग्राम हो, और कुछ वाइल्ड वेराइटीज भी होती है, वो सभी वेराइटी जो है ये वाइल्ड जो ट्राइबल पीपल्स है वो उसको संरक्षित करते हैं। कुछ ट्यूबर्स है जो जंगल से कलेक्ट करके एक स्पेस में रखते हैं। अभी कई जगह पे तो एनजीओस आ गए हैं जो एनजीओ उनको स्पेस देते हैं, वहां पे वो ट्यूबर जो है उसको संरक्षित करते हैं। और कहा था कभी कभी उनको जो ये सीड्स है, ट्यूबर्स है, इनको वर्नेकुलर पता है, बट बेसिकली जो रिसर्चर है उनको ये वाइल्ड वैरायटी उपलब्ध करने के लिए इनके पास से एनजीओ कलेक्ट करके उनको लेबलिंग करता है, जिसको एक सीड बैंक के तहत, और उन सभी जो वैरायटी को संरक्षित करता है, देन उसको आइडेंटिफाई करता है। इम्पोर्टेंस ऑफ सीड बैंक, जनरली ये जो है एक तो स्टैंडर्ड फॉर्म में लिख रहे हैं हमारे लिटरेचर के फॉर्म में, बट बेसिकली जो ट्राइबल पीपल्स है वो संरक्षित कर रहे, उनका एक ही होता है। जब लॉकडाउन का समय था उस समय उनके पास लगाने के लिए वैरायटी, उनको शहर में जाके वो लाना बीज के जो सेंटर्स थे वो क्लोज थे, उस समय उनके पास की खुद की जो सीड बैंक, उस सीड बैंक के सीड जो थे वो अपने खेती में आपल्या घरच्या वाड्यांमध्ये त्यांनी लावलं त्यावेळेस. दैट मीन्स जो उनकी सीड बैंक पर्सनल है, वो सीड बैंक सचमुच एक आदर्श है जो हमारे को बताती है कि हमारा जो ओल्ड इज गोल्ड हम कहते हैं, इट्स ओल्ड बट इट्स अ गोल्ड. तो ये एक वर्ड है जो ट्रूथ जो है एकदम सच्चा है. तो बेसिकली सीड बैंक की इंपॉर्टेंस यही है, प्रिजर्वेशन ऑफ क्रॉप डाइवर्सिटी. डाइवर्सिटी को हम इसमें प्रिजर्व कर सकते हैं. कुछ लॉस्ट हो रही है वेराइटीज, उसको हम प्रिजर्व कर सकते हैं, संरक्षित कर सकते हैं. प्रोटेक्शन फ्रॉम क्लाइमेट चेंज, हर समय हर साल क्लाइमेट चेंज हो रहा है, उस हिसाब से कुछ वेराइटीज है, कुछ वाइल्ड वेराइटीज लॉस हो रही है, उसकी सीड्स हम प्रोटेक्ट कर सकते हैं. देन प्रोटेक्शन ऑफ नेचुरल डिजास्टर, बहुत सारे हर साल नेचुरल डिजास्टर आ रहे, उस वजह से भी फॉरेस्ट है, फॉरेस्ट पैच है, उसके बाद कुछ जो है रेसिडेंशियली जहाँ पे वेराइटीज है वो डिस्टर्ब हो सकते हैं, वैसे सीड्स को हम संरक्षित कर सकते हैं. डिसीज रेजिस्टेंट जो मैं बोल रहा हूँ, उनकी मेथड है ट्राइबल पीपल की, लाइट एंड वेटेड सीड्स है उसको क्लासिफाई करते हैं, और जो डॉर्मेंसी नहीं होती है वैसे सीड को वो लगाने के लिए करते हैं. तो रेजिस्टेंस ऐसे सीड है, वो डिसीज रेजिस्टेंस क्रॉप जो है ये भी हम संरक्षित कर सकते हैं. उसके बाद रिसर्चर को हम ये सीड एज अ मटेरियल करके प्रोवाइड कर सकते हैं. देन द प्रिजर्वेशन फ्रॉम मैन मेड डिजास्टर, जो हमारे एक्टिविटी उस वजह से बहुत सारे डिजास्टर होते हैं, उस वजह से भी बहुत सारे वाइल्ड वेराइटीज, लोकल वेराइटीज एज वेल एज क्रॉप जो है इसकी खत्म हो सकती है, वो भी वेराइटीज एज अ फॉर्म हम जो है संरक्षित कर सकते हैं. थैंक यू सो मच. बेसिकली ये मेरा वर्क नहीं है, बट मैं पर्सनली बिचवा पातेर एक प्लेस है चांदना माटा पराशिया के आगे, वहां पे विजिट किया था, तो मैंने उनकी एक सीड बैंक देखी थी. ये फोटोग्राफ मैंने नेट लिए, बट ये कंसेप्ट मुझे बहुत पसंद आया, इसलिए मैंने आपके नोट ऑन सीड बैंक ये आपको बताया है. थैंक यू वेरी मच मैम. थैंक यू वेरी मच सर.
थँक्यू डॉक्टर संजय सर. नवीन कन्सेप्ट गोष्ट तुम्ही सांगितली. थँक्यू सो मच. डोंगरवार सर काही विचारायचं आहे?
नाही नाही मॅडम, काय
ओके. नेक्स्ट
आणखी
पार्ट
कोणाचा आहे आता?
सर नेक्स्ट आहे खुशल बोरकर. डॉक्टर खुशल
अच्छा,
बोरकर.
खुशाल. डॉक्टर
या
खुशाल.
खुशल सर, आप अपने तरफ से प्रेजेंट करने वाले हैं ना?
यस मैम.
हाँ,
यस
yes.
मैम.
Just a minute. I'll make you presenter. Yeah. Now you can share your screen. Yes, sir. Please
Good
start.
afternoon, everyone. Good afternoon, everyone. Respected Dongarwar sir, in charge of the refresher course, and Dr. Priti Dharmik madam, coordinator of the course. I am Dr. Kushal Borkar, presenting my seminar topic on thermophilic fungi, their physiology and enzymes. This is the content of my seminar. Then, among the approximately 2.2 to 3.8 millions of fungi present over the earth, only 1.2 lakh fungi are accepted, and the thermophilic fungi have a very small assemblage in them. Pune and Emerson in the year 1964 formulated a less elaborate definition as the thermophilic fungal species can be defined as those with the minima for growth at or above 20 degree and maxima for the growth at 50 or above, whereas the thermotolerant fungi are ones that have the thermal maxima near 50 and minima below 20. So this is a definition of thermophilic fungi, and from around approximately 1.2 lakh recorded species, thermophilic fungi have very small assemblage. And depending on the temperature tolerance, fungi can be categorized as psychrophiles, those grow below 20 degree, then mesophiles 20 to 45 degree temperature range, and the thermophiles 20 to 60 and above range. Perhaps because of their moderate degree of thermophily, and because of their habitats are not so exotic, thermophilic fungi have not been received much publicity and attention. And one extremely valuable advantage of conducting work at the elevated temperature is reducing the risk of contamination by common mesophiles, because this fungi has to be isolated and has to be studied under, at 45 degrees centigrade Therefore, the other mesophiles do not get contaminated with these fungi. That's why the work on this thermophilic becomes easier. Then the thermophilic fungi is nothing but it's a ecological group depending on the temperature range, because we know that there are so many fungi. We call them depending on their habitat. We call them as a soil fungi, aquatic fungi, coprophilous, entomopathogenic, predaceous, marine, osmophilic. Likewise, these thermophilic fungi are dependent on the temperatures: thermophilic, mesophilic, and psychrophilic fungi. Then coming to the habitats of these thermophilic fungi, they have worldwide in distribution, mostly in the self-eating masses. Like they are present in the soil, the air, hot springs, then nesting materials of birds and animals, piles of hay, then compost, which is a self-heating mass, then coal spoil tips, where the surrounding soil gets heated, then herbivores dung, wood chip piles, then municipal refuse, then peat, and also in the stored grains. So these are some of the habitats for thermophilic fungi. Then coming to the applications of thermophilic fungi, how these thermophilic fungi are important. Single cell protein can be isolated from this. Soil fertility can be increased. Then the thermostable enzymes are very much important in the industrial point of view. Then the antimicrobial and other bioactive compounds can be isolated. Biotransformation and bioremediation experiments can be done. Then the composting process, they bring about the composting process in very faster manner than the biofuel production. These fungi can be utilized for the biofuel production also. So these are the applications of thermophilic fungi. Then coming to the methods for the isolation of thermophilic fungi, when one has to bring the sample, and that sample has to be activated in incubator at 45 degrees centigrade for 4 to 5 days. Then the sample has to be taken on the taken by direct plate method by the suggested by Warkup in 1950. Then activated samples will be directly inoculated on YPSS agar medium by direct plating method. Then these plates will kept in the incubator at 45 degrees centigrade for incubation. After 2 to 3 days, fungal colonies will be seen. Then pure culturing for identification and further use will be done. Then in the further experiments, temperature requirement studies, similarly the pH requirements, nutrition requirements, radial mycelial growth, mycelial dry weight studies can be done. Then these fungi can be grown on the agricultural waste to degrade them at the faster rates. And while degrading, we can study their enzyme activities, and also the further experiments can be done with these fungi. Then during the isolation, this much list of the fungi along with others can be seen, where there is Rhizomucor pusillus, Rhizopus microsporus, Rhizopus rhizopodiformis, Emericilla nidulans, Critomium thermophile, Myriococum albumyces, Thermoascus orientaceus, Aspergillus fumigatus, Malbranchia pulchilla, Sporotrichum thermophile, Humicola insolens, and Thermomyces lanuginensis can be seen on the culture plates. Then these are some of the micro photographs of fungi, where this is Rhizomucor pusillus, this is Rhizopus rhizopodiformis, this is Emericilla nidulans, this is Chitomium thermophile, this is Myriococum albumyces, Thermoascus orientaceus, this is Humicola insolens, and the Thermomyces lanuginosus. Then this is the table showing the list of list of fungi recorded, and their optimum temperature as well as maximum temperature is shown in this table. If we consider the 45-degree temperature, we can see that the 45-degree temperature as optimum can be seen for most of the fungi, whereas in some of the fungi, the 50-degree centigrade temperature is also the optimum. In some of the fungi, the maximum temperature is 60 to 61 degrees centigrade. This is the growth pattern of Chitomium thermophile at different temperatures. The temperature range for this is 30 to 55 degrees centigrade, and The plot is against radial mycelial growth in centimeters against the days of incubation. From 0 to 8 days, these readings were taken, and 45 to 50 degree centigrade temperature was suitable for their growth. And in the 50-degree temperature was giving the rapid growth, and it was completing its growth within 4 days. Growth patterns of chaetomium thermophile at different pH. This graph shows the growth pattern of chaetomium thermophile at different pH. The pH range of 4 to 10 level was given, and 5, 6, and 7 pH was supportive for the growth, which was completing in 4 days. These are the photograph. This photograph shows the enzyme activity, carboxymethyl cellulase studies on different substrates. Substrates were taken from different sources, and the fungi were also different for the study of crude enzyme extract as a CMCH activity. Then these fungi yield industrially important thermostable enzymes. Thermostable means they are stable at the high temperature. Some proteases, lipases, amylases, cellulases, xylanases, lactases, and phytases are reported by many workers from the thermophilic fungi. These thermostable enzymes are very famous and industrially important because they give a reduction in the enzyme needed for hydrolysis, enhance recovery of volatile compounds, decrease in the reaction time, increase rate of reaction, then decrease contamination risk, lower viscosity, and longer half-life. These are some of the characters given by thermostable enzymes. That's why they are very famous from the thermophilic fungi. Presence in coming to the conclusion, presence in most of the habitats, preferably in the self-heating. Studies on the physiological parameters gives idea for further experimentations. Minimum temperature is 20 and maximum up to 60 degrees. Enzymes tolerate variable ranges of temperature and pH requirements. The extracellular enzymes of thermophilic fungi are appreciably thermostable. The comparison of properties of enzymes from 2 or more strains by the same method will be necessary to determine if high variability is characteristic feature of thermophilic fungi. 2 different species can be studied for the same source of enzyme, and the same can be studied for the variations. Then if so, investigations of the mechanism generating variations may provide opportunity for the fundamental discoveries. Modern studies on thermophilic fungi were stimulated by the prospect of finding fungi capable of secreting high level of enzymes and of the finding novel enzyme variations with high temperature optima and long shelf life, which are desirable characteristics for commercial applications of enzymes. These fungi have potential to be used in the genetic engineering as a source of thermostable enzymes. And these are some of the references used for making presentation. Thank you.
Thank you so much, Dr. Kushal. Sir, any question?
Yes, there is one question for Dr. Kushal.
Yes, sir.
Dr. Kushal, can we use Waxman method for identification, because you have mentioned only direct plate method? So
yes, sir. Other methods also can be used, but direct plating method gives a better result. That's why I said this one is the good method.
Yeah. Thank you.
Other methods also can be used. Yes, sir.
Okay.
Hello, sir.
Are there any specific media, or we can use only Zapec docs or
Sir,
Podadodextrose
actually,
agar?
YPSS agar medium is a special medium for isolation.
Okay.
YPSS agar medium is special for isolation.
Okay. Thank you.
Okay. Thank you, sir.
Hello, sir.
Yes, ma'am.
Very nice work. I just wanted to have your view on whether we are using such fungi, that is thermophilic fungi, for plastic degradation. So what do you think? Will it work on it?
Ma'am, the work is going on in world over for the degradation, but not getting some good results as compared to other organisms, which many workers have reported. Not some good results are present there.
Yeah, but like I wanted to know your view. Like it is producing so many types of enzymes. So is there a possibility? Can we check on it?
Maybe some fungus if you isolate and it degrades the plastic, there is a possibility in the further research.
Okay.
If you conduct some research,
Very
there
less
can be.
reports of fungus are there, more of bacteria are there. So I
Yes.
just wanted to have your view. Yeah, fungi reports are very less.
Yes,
I
ma'am.
wanted to have your view on that.
Okay.
Okay, sir.
Thank you, sir. Now our last presenter is Dr. Machindra Nandeshwar, sir.
Yes, yes, ma'am. I am here.
Yeah. PPT दिसता आहे?
हो.
Yes, please start.
Good afternoon, respected Dr. Dongarwar sir and Dharmik madam and everyone participant. I am Dr. MP Nandeswar. My topic is the history of life. Next. Here is fundamental character. Just we know here is big idea. All living things have certain traits in common. Cellular organization, the ability to reproduce, growth and development, energy use, homeostasis, response to their environment, and the ability to adapt. There is the energy acquisition and utilization, information storage, reproduction, evolution by natural selection, these all things. Next, madam. Disappearing the earliest history of the earth, zircon grains. Just we know that the earth was early one very hot sphere and some activities happened inside the sphere like bombardment and some chemicals, gases and some other that materials come from the earth and reactions takes place in nature. That's chemical reactions, gases reactions, and uh, then such type of zircon grains. And here is the evolution to acceleration disk farm to some differentiated asteroid, Mars acceleration, moon farm, till to end of intense bombardment. Next, madam. Then zircon grains are also formed, that is common mineral zirconium silicates occurring in small tetragonal crystals or grains of various color, usually opaque. Next slide. How extraterrestrial origin of life, panspermia. Next slide. Murchison meteorite. What is this? The Murchison meteorite is a meteorite that fell on Australia in 1969. In this picture, you can find there is Murchison, and this meteorite fall there near Murchison, Victoria. It belongs to a group of meteorites rich in organic compound. Due to its mass and the fact that it was an observed fall, the Murchison meteorite is one of the most studied of all meteorites. Next. Evidence of life on Mars. So, to date, no proof of past or present life has been found on Mars. Cumulative evidence suggests that during the ancient suggests that uh, uh, Nochian time period, the surface environment of Mars had liquid water
and may have been habitable for microorganism, but habitable conditions do not necessarily indicate life. But here is one picture is that are these biological microfossils just like we see in microscope. There is look like as a microfossils. Next slide. There is a big unanswered questions in origin of life research. How did the primordial soap acquire the simple monomeric building blocks essential for the production of information bearing polymer? What conditions are necessary for the initial prebiotic assembly of such polymers? Can a polymer be produced that is capable of self-replication as well as information storage? How did compartmentalization necessary for recognition during replication and for the diffusion of gene product evolve? Which came first? This is the question. DNA, RNA, protein, or something else? Or did complex system involving all of these emerge simultaneously? Next slide. This experiment, everybody familiar about that. This experiment that is Urey Miller's experiment. We learn this experiment. Next, madam. Assembly of an information bearing polymers. Here is protein first. There is a confusion. There is a 2 points is here, a strong point and weak point. The strong point is that easy to synthesize amino acid under a variety of condition and polymer can also be formed. Even small peptide can exhibit catalytic activity. The 20 amino acid provide for high information content. Weak point is that globular structure and lack complementary preclude self replication. A modern protein cannot function without DNA. Next slide. And RNA world. Strong point is that RNA has some catalytic properties, that is self-splitting introns. RNA is capable of making protein, and weak point is RNA lack the ability to self-replicate. A DNA word. DNA is almost completely lacking in catalytic ability. Next slide. The emergence of cell during the origin of life. It appears that life first emerged at last 3.8 billion years ago, approximately 750 million years ago after Earth was formed. This figure, observation in this figure, there is a genome replication, then membrane transfer, then division occur. The emergence of cellular behavior, competition emerges as protocells containing replicating genome, still membrane form protocell containing inactive molecules. Next. Evolution of early life form. Next. Just the Charles Darwin, what type of ideas used by Charles Darwin, and everybody known about that. The Charlene said, if the theory, that is evolution theory, be true. Next slide, madam. Hello. If the theory of evolution be true, it is indisputable that before the Cambrian stratum was deposited, long period elapsed, and that during this vast period the world swarmed with living creature. However, to the question why we do not find rich fossiliferous deposits belong to this earliest period, I can give no satisfactory answer. The case at present must remain unexplicable. Next, madam. This is ancient prokaryotes from Western Australia. This found in Western Australia. This is the cyanobacteria. It is filamentous cyanobacteria found 3.5 billion years ago. Next slide. Earliest filamentous microfossils. These picture shows these are the microfossils. Next slide. Microfossils, these picture shows the cyanobacteria. Next slide. And these are stromatolite from Western Australia. Next. Then, after a study how the many eukaryotes are found on Earth, the evolution state, the origin of eukaryotes, there is earliest probable eukaryotes are single-celled algae. These found in 1.6 billion years ago. And definitive evidence for the eukaryotes exists from about 1.2 billion years ago in form of fossils for multicellular algae. Next slide. How the evolution takes place in a sexual reproduction. Madam, next slide. Next slide. See here, the primitive eukaryotes, that is Gardia lamblia. This is the microorganism, and these have a two haploid nuclei. There is a absence of mitochondria. Next. Evolution and diversification of the eukaryotes. What factor contributed to the rapid diversification of eukaryotic lineage? That is increased atmospheric oxygen concentration, switch to aerobic respiration, global climate change, major ice age around 2.7 billion years ago, evolution of sexual reproduction. Just we see in Jurassic movie, there is a very large animals, very large trees due to just newly formed atmosphere, and there is a huge atmosphere very यानी उसको बोलते हैं कि सुटेबल एटमॉस्फेयर था, इसलिए वो उनका अच्छा हेल्थ था. नेक्स्ट. थैंक यू.
थँक्यू सर. सर काही क्वेश्चन्स आहेत का?
नाही मॅडम.
ओके.
झाले का आजचे सगळे?
हो सर, आजचे संपले. सर तुम्हाला काही इन्स्ट्रक्शन्स द्यायचे असतील तर?
फक्त एकच इंस्ट्रक्शन है सभी पार्टिसिपेंट्स को, सिर्फ एक ही इंस्ट्रक्शन है, वो है कि हम जो है समय का पालन करें, और जब कोई प्रेजेंट कर रहा है तो दूसरे लोग इस चीज़ का ध्यान दीजिए कि आपका माइक गल
हेलो
हॅलो.
सर
आवाज नहीं आ रही थी.
सरांचा आवाज नाही येत आहे ना?
नाही ना. प्रीती मॅडम.
ओके. हां सर.
हां प्रीती मॅडम, मला प्रेझेंटेशन करायचं असल्यास आज तर वेळ निघून गेला नाही का?
सर आता आपलं टाइम टेबल शेड्यूल आहे ना, त्याच वेळेस उद्या
हो हो.
उद्या बॉटनी झुलॉजीचे आहे. आय थिंक तुमचं
अच्छा.
सॅटर्डेला आहे.
माझं सॅटर्डेला
तर
लास्ट डे.
सॅटर्डेला मॉर्निंगला आपण 10 वाजता सुरू करूया प्रेझेंटेशन्स.
चालेल ना, चालेल.
ठीक आहे.
नाही असं तुम्ही म्हणलात ना, कधी कुणाला
हां,
जर
जर लवकर झालं तर.
रेडी
पण
असेल.
आज ऑलरेडी आपला वेळ झालाय ना,
हां,
म्हणजे तेवढा
हां.
वेळ लागतोच
राइट,
आहे
राइट.
सगळ्यांना प्रेझेंट करायला. तर
नाही,
माझं
नाही
हेच
प्रश्न
होतं,
नाही.
की
10
जर थोडं
मिनिटं.
लवकर झालं 15-20 मिनिटं तर एखादं प्रेझेंटेशन आपण ॲडिशनल घेऊ शकलो असतो. पण
हो,
वेळ
हो, हो.
झालेला आहे
हं.
कंप्लीट. जर
नाही,
उद्या
नाही काही
झालं
इश्यू नाही, काही
लवकर तर मग उद्या घेऊ आपण. तुम्ही रेडी राहा. जेव्हा होईल कंप्लीट तेव्हा आपण
हो, मी आज
घेता
पण
येईल.
वाटच बघत होतो. म्हटलं
पण
एखादा
लागलाच वेळ होतोच. 10 मिनिटं जास्तीच झाले
नाही, नाही
तुझे
काही
पण.
काही इश्यू नाही, काही इश्यू नाही. पण छान
ठीक आहे.
प्रेझेंटेशन देतात आहे सर्व. छान. ओके
ओके,
मॅम.
ओके.
थँक यू.
चलिए, थँक यू ऑल ऑफ यू. सरांचा तर आवाज येत नाही आहे. So I'll thank all the participants and see you tomorrow at 6:45. Thank you.
थँक यू मॅडम.
Thank
Thank
you.
you, madam.
थँक्यू मॅम.
And congratulations to all for nice presentations.